and CURE's mission and a little bit about the seminar series. So, Steve?
Thanks, Karen, and thank you all for your attendance this afternoon. And it really is an honor to sponsor Dr. Lowenstein for this fifth in a series of seminars that CURE is sponsoring this year, Frontiers in Research Seminar Series. And the goal behind this seminar series is to introduce those who may not be familiar with epilepsy research into epilepsy. Also to bring in preeminent researchers and scientists in the area of epilepsy research to discuss ongoing activities and the state of the art in epilepsy research. And as you'll hear in a moment by Karen, Dan certainly meets that criteria, and we're so delighted that he accepted the invitation. CURE was founded in 1998 by Susan Axelrod and a number of research grants around the world, and many of these have led to some important breakthroughs in how we're approaching epilepsy. And to that point, I think it was the voice of Susan and others at the very first Curing Epilepsies conference in 2000 that changed the scope and changed the face of how we talk about epilepsy research today. We're not only just talking about the symptomatic treatment of epilepsy, but we're talking about the possibility that a cure may really be out there if the right resources and focus is aimed at finding it. And so CURE is very proud as the largest non-governmental funder of epilepsy research to be able to help lead that forward. And to that point, I put on the back table back there a little information about CURE. And on the back page, it describes the funding mechanisms. We have 6 funding mechanisms available. We would like to encourage those that might be interested to submit LOIs. We have 2 LOI deadlines each year. And those are reviewed and peer-reviewed. I would encourage you to look at that. If you do have questions about CURE and its mission, I'm on campus. I'm happy to answer any of those, or you can reach out to the CURE offices as well. So with that as an introduction, I'll turn over to Karen, who will introduce today's speaker.
Very much, Steve, for that great introduction about CURE. And really, when we were asked to select a speaker for our part of the seminar series, it took Steve and I about maybe 5 milliseconds before we figured out that Dr. Dan Lowenstein was the person we wanted to come visit us today. So I want to tell you just a little bit about Dan and his work to date before he talks to us. So Dan is currently the Vice Chairman and Professor of Neurology and Director of the UCSF Epilepsy Center. He's the Director of the Physician Science Education and Training and Associate Dean of Clinical and Translational Science at UCSF. He graduated from the University of Colorado with a degree in mathematics and received his MD at Harvard Medical School. He then did his neurology residency at UCSF and a 2-year molecular biology fellowship in Stanley Prisner's lab. And he went on to become the Robert B. and Eleanor Aird Professor of Neurology and established the Epilepsy Research Lab at UCSF. He served for a while as Dean of Medical Education at Harvard from 2000 to 2002, where he oversaw a major overhaul of the curriculum there. And then he returned back to the Bay Area to renew his position at UCSF. Dr. Lowenstein is a clinician scientist who has studied both science and basic science and clinical aspects of epilepsy. His laboratory studies have examined the fundamental basis of neuronal network remodeling that occur during epileptogenesis, and with particular focus on the parallels between injury-induced remodeling and the molecular and cellular mechanisms underlying normal brain development. His clinical research includes studies on the management and treatment of patients with status epilepticus, and he is now the principal organizer of a large-scale international effort he'll be talking to us about today. He has received numerous awards, many awards, too many to list here today, but some of the highlights are from the American Epilepsy Society. He received in 2001 the Basic Research Award, and he was the 2012 recipient of the AES Lennox Award as well. He also received this year the Ambassador Award from the ILAE, or the International League Against Epilepsy, in recognition of outstanding international contributions to the cause of epilepsy. Dr. Lowenstein, and this is one of the reasons we also wanted to bring him here, is an avid and highly regarded teacher, and he has had an active role at UCSF concerning issues related to cultural diversity as well as civil rights. And in recognition of his leadership in this regard, he's received the 1998 Black Student Health Association's Faculty Award, the 1998 UCSF Dr. Martin Luther King Jr. Award, the Chancellor's Award for Public Service as well. And he recently was selected to deliver the last lecture at the UCSF community, which was luckily not the last lecture. It was a very prestigious series of seminars. And Dr. Lowenstein has been actively involved in advancing the cause of epilepsy at the national and international level as well, and has had numerous leadership roles in professional organizations and foundations, including serving as the president of the American Epilepsy Society and chairman of the Epilepsy Benchmark Stewards. And on a personal note, he and I have served as co-chairs together on the Translational Symposium of the American Epilepsy Society, and I can tell you it was an absolute delight to work with Dan on that effort. So it's a great pleasure for me, in particular, to introduce Dr. Lowenstein today, and he will talk to us about large-scale team science and epilepsy genetics, the EPGP and Epi4K experience. Dan?
Thank you. Thanks, Karen. Thanks. I appreciate it. Great. Well, thank you for the introduction. Can you hear me in the back okay? Sound about right? Good. Truly a pleasure. I greatly appreciate the invitation. I've had a wonderful, very full day. Particularly liked hanging out with the grad students over lunch, hearing about what you're doing and exchanging some stories. And it's particularly nice to be at one of the important institutions that has been at the vanguard of advancing our knowledge about epilepsy and from basic mechanisms to clinical aspects. So it's a real treat. I wanted to recognize a whole host of organizations led by people who have been absolutely critical to the progress that all of us are making. And you'll notice that CURE is headlined right at the top. I've been very fortunate to get to work with Steve on CURE-related projects, but there are a whole host of funding agencies and forms of support that have helped the work that I'll be describing today. And so here's what I'd like to do over the course of about an hour, 50 minutes or so, give or take, and I can skip through sections as needed. But I wanted to cover the following. First of all, I assume that there would be a fair number of people attending this discussion who don't participate directly in the care of patients on a day-to-day basis. So I wanted to talk a little bit about the reality of living with epilepsy. Then a brief discussion about what we know about the genetics of epilepsy just from a historical perspective to make sure that everyone realizes that there's no question that genetics plays an important role. Then an equally brief overview of epilepsy gene discovery to date. And then I'll focus in on the story of EPGP, I'll call it EPGEP, the Epilepsy Phenome Genome Project, and Epi4K, the latest project that a large number of us are working on. And I'll try to sort of emphasize some of the themes and lessons that we've learned, both some of the core science that's come out of recent discoveries, as well as the process of putting together scientific enterprise of the scope that you'll realize has occurred with EPGEP and Epi4K. Okay, so that's the plan. So let's start with the reality of living with epilepsy. I base this set of observations on the experience that any of the clinicians in the room who take care of patients with epilepsy will have. I have an epilepsy clinic that I've attended half a day a week for now many years. I take care of a couple of hundred patients over the long term and see many new patients every week. And I just picked two examples of one particular Tuesday afternoon now a few months ago, and I wanted to describe them. The first is a 17-year-old student. She was brought in with her parents. She was a high school student at Tam High in Mill Valley, California. And the three of them came in because she had had some recent concerning events. She had a longer-term history of these things that they called night terrors since childhood. And then over the previous year, she had 2 episodes of frightened behavior followed by focal clonic activity and secondarily generalized tonic-clonic seizures. So that was her story. And I don't need to go into the details. There are plenty of details that are relevant to hearing about a new description of unexplained spells. But the reason that I highlight this story is that it was obviously an incredibly concerning event to occur to a family. I think this patient had a sister, so it was a family of 4. But here was a very, very high-functioning high school student destined for college this year, and these events occurred. When they came to see me, they came with what I call the universal questions, the 3 universal questions that all of us ask of our healers, of our physicians, our healthcare professionals. No matter what it is that's wrong, if we go and seek someone's help, there are 3 questions. The first, what's wrong with me? What's happened to me? The second, can you help me? Is there something that you can do that will help me, that will help us? And the third, what does our future hold? What does my future hold? Right? Think about that. Anytime you've gone to see someone, whether it's a sprained ankle or it's something you have no idea what it is, those are the 3 questions. You know, what's happened? Can you help me? And what does my future hold? You can imagine what that must have felt like for this family. And in the long discussion that we had after looking at the information and concluding that it was highly likely that this young woman has epilepsy, probably of a genetic background, that I couldn't answer any of those questions fully. What's wrong with me? Well, I could explain to the family, based on the information, that the epilepsy probably has a complex genetic type of cause. But I wasn't sure. Certainly no test that I could send off and provide an answer. Can you help me? Well, actually plenty of the people in the room know this very well. There are lots of different medications that can be offered to try to prevent the recurrence of these episodes, in addition to lifestyle changes and so forth, to be careful. But do I know for sure whether the drug that we pick together, is going to be the drug that prevents having any further seizures soon or later? And will there be any side effects from the medication that we're providing? Again, you can imagine knowing about the toxicity profile of essentially all the medications that we offer, and that's not just anti-epileptic drugs. Very, very high-functioning a young person hoping to get into UC Berkeley, she would say, we can't afford to have anything that's going to take away my level of energy or my ability to concentrate or my overall ability in cognition. So I could tell them that we do have medications. They can be very, very effective. But in terms of being certain whether or not it's going to be useful, I couldn't tell. And then finally, what does my future hold? Honestly, I have no idea. Does anyone know what this person's future holds in this room? No way. We have no bioassay. We have no way of measuring the activity of the underlying disease. So will I ever have a seizure again in my life? I don't know. Will my epilepsy ever go away? Does it ever go away for people? Yes, it does, but in your case, I'm not sure. When will that happen if it does? No idea. Okay? So let me tell you about the second patient. This is a 16-year-old young woman who I actually got because she was referred to me from Orrin Davinsky, a very close friend of mine at NYU, about 8 years earlier. 10 years before, she had undergone a temporal lobectomy at a relatively young age. As a 6-year-old, that's really young. The temporal lobectomy took care of her seizures. She had a terrible seizure burden beforehand. And she had the lobectomy, and she did incredibly well. We kept her on, I believe it was lamotrigine, for a number of years. And by the time she was maybe 13 or so, we decided it was safe. She was completely seizure-free. And we decided to begin to taper her off her lamotrigine. And over the course of about 6 months, we very gingerly took her off the medication. And she remained seizure-free. And we were celebrating, the two parents and the patient and I, when we'd see each other every 3 to 4 months, continued to be seizure-free off medications, really regaining her headway in high school, doing incredibly well. And then, a few months ago, actually this is now a number of months ago, I got a desperate call from her mom. I won't name the patient. I'll make up the name. Mary apparently had a fainting spell 6 weeks before. And when we went over the history, when they came in, it was uncomfortable to sense that this fainting spell sure sounded a lot like the focal seizures with dyscognitive features that she had had many, many, many years ago. In fact, it came out that maybe she was having some small events that she actually didn't pay much attention to because they weren't disrupting her life. And I can tell you that since this event, she's now had 2 convulsive seizures of focal onset. So her epilepsy actually was never cured by the lobectomy, and now we're facing the prospect of being back on lamotrigine and the uncertainty that carries. So again, sorry, is that you? Got it? Okay. Just want to make sure it wasn't my phone. And so again, the same issues. What is wrong with me? Why is it that we've removed the focus, the temporal lobe focus, and now the epilepsy has come back? What can you do? What can we do for her? Do we know whether the lamotrigine is going to continue to be effective? So far, it looks as though it has, although she's had at least one description of what I'm concerned is a simple focal seizure. And then what does her future hold? I have no idea. So this is the lowest part of the talk, okay? Everything's going to get a little bit better from here. This is so frustrating. It's so discouraging to realize that so many people with epilepsy face these kinds of experiences. This is the reality of certainly at least 30% of all people who have epilepsy. And it extends into that group that we call controlled with medications, because many of those individuals certainly will describe side effects. So our goal of no seizures and no side effects is actually not achieved in a significant number of people with epilepsy. And as I mentioned briefly to the folks last hour, I don't know for those of you elsewhere in the university, but the anticonvulsant drug development program is one of the shining stars in this country in terms of discovering the treatments that we absolutely need to have in order to change these kinds of experiences. It's only going to be through the kind of focused, dedicated work, both from a fundamental mechanistic level, as well as cranking out the studies and screening drugs in a variety of models, that we're going to be able to come up with the medications that are going to allow us to treat patients who have seizures, both in terms of fully controlling their seizures without side effects, as well as discovering those approaches, those drugs that can prevent epilepsy in the first place, the anti-epileptogenic compounds. So I hope you folks appreciate how lucky you are to have such an important program here. That's what patients are like when they come into clinic. Just to underscore the impact in terms of what living with epilepsy is like, patients face the unpredictability of seizures, not having any idea when they'll tend to occur, actual real significant risk of injury and even death from having recurrent seizures, all sorts of comorbidities, restrictions on driving, problems in terms of trying to get educated and having a job, lack of independence and stigma. A huge number of people around the United States and the world. The latest statistic, 1 in 26 individuals, 1 in 26 humans have developed epilepsy, and a huge cost. And if you ask patients, what's the worst thing about having seizures? This is something that Bob Fisher at Stanford published back in 2000. These are the percent of responders who described these sorts of concerns. And of course, the leading one was fear of having another seizure, but then problems with anti-epileptic drugs, limitations to their life experiences, stigma, and so forth. So let's go to the second topic. How important is genetics as a cause of epilepsy? So let me just touch briefly on some observations that relate to epidemiological studies. And the first is that we know from work done out of Rochester, if you look at the population, the overall population in a region, and identify those patients who have various epilepsy disorders and try to categorize them, that some of them can be ascribed to things like a stroke or trauma or a congenital abnormality that occurred in utero or other so-called acquired events. Or the embryo was presumably normal, and then some environmental or external event occurred, and the person acquired epilepsy. But the vast majority are called unknown or idiopathic. Now, two studies, just again, this is mostly for a historical point of view, but it's for the students in the room to make sure that you understand why it is that we know that genetics plays an important role. So the first is an absolutely classic study that was published in 1947 by William Lennox, one of the giants in the world of epilepsy during the last century. And Lennox had a very busy practice, saw like a gazillion people with epilepsy, and he was able to collect a large number of twins, 66 pairs that he described, and studied the nature of their phenotype, both in terms of their clinical presentation and their EEG, and did the classic twin study. You look at concordance between the dizygotic and the monozygotic twins. So here's one example. This is Constance and Catherine. Some of you may have seen this sort of legendary figure in the past. So there they are. They look pretty similar. And here's their EEGs. Also looks pretty similar. And they had a 3 per second spike and wave form of epilepsy. And this was the main result from Lennox's study. And this shows the proportion of twin pairs in which either they did or did not have injury. So Lennox understandably wanted to divide the groups into those that had no obvious acquired event in their life, like a traumatic brain injury, to those that did. And then he looked at the monozygotic versus the dizygotic. And as you can see, for those in which there was no prior identifiable external event, there was significant concordance only among the monozygotics, up to 83%, as opposed to the others. So this shows very clearly that within twins, genes play an important role. Now, the other classic study came also out of Rochester. This is by Anagers et al., published in 1982. And this was a population-based study. In this case, they looked at the cohort of patients who had epilepsy and inspected the entire family structure to look at the relative risk of developing epilepsy if a family member had epilepsy. So to do that, they identified 196 cases of idiopathic epilepsy. Onset was between birth and 15 years. And then another 60 cases of isolated seizures, again from this area, from Rochester. And this is the design of the study. This would be one of those 196 individuals in the population who had the diagnosis of epilepsy. Once that proband was identified, they then attempted to build the family tree, both upward and downwards. So in order to be enrolled in the study, they needed to be able to identify and make sure what the epilepsy history was in both parents. And then they traced the family tree and then asked the question, based on this proband, what was the relative risk of having epilepsy if you were born to a parent with epilepsy, or you had a sibling or a nephew, and so forth. Okay? So that's the design, and these are the main data. And what this shows is the relationship between the individuals, the observed cases, the expected cases just based on the occurrence of epilepsy across the population at large, and then they calculated the relative risk. And what you can see is that a clear significant effect was seen in the individuals who had a sibling with epilepsy. So if you had a brother with epilepsy, you were at 2.5-fold increased risk if you were that sibling, another brother or sister. In addition, if you had a mother with epilepsy, as a child, you had a 6.7-fold increased risk of developing epilepsy. So again, additional very important and compelling evidence to show that there is a genetic effect in the epilepsies writ large. Okay, so third topic, a brief overview of epilepsy gene discovery to date. Well, this is kind of fun to be able to show this, part of the discussion here because I feel like I'm like at the homeland. It kind of all started here. So in this landmark paper in 1989 in Nature that Mark Leppert, Ray White, and colleagues published showing a clear genetic linkage to one particular epilepsy syndrome, benign familial neonatal convulsions, linked to chromosome 20. Back in those days, I'm sure I haven't seen Mark recently, but I'm sure he'd wonder how long it would take to find the gene mutation if they had this linkage today. Probably a matter of a week or something. It took about 9 or 10 years, I think, and that's just the way science goes. But anyway, an incredibly important paper and really set the stage for the discoveries that followed. The first actual identification of a true epilepsy gene mutation came out in 1995 when Steinlein and Berkovic and others described a mutation in the neuronal nicotinic acetylcholine receptor subunit. And this was based, as was the case for most of the gene discoveries at the time, on incredibly interesting and informative pedigrees, such as this one. This is an Aboriginal Australian pedigree in which clearly there's Mendelian inheritance, and using the techniques at the time, they were able to identify this particular mutation. And here's a list, not complete, and it is an incomplete list of epilepsy genes as of now. Definitely, this is definitely one of those slides where I know I'm supposed to say, and I'm going to say, this is not meant for you to read all the detail. The whole point is just to know that there's a big list. But I did want to highlight a couple of things about this. So first of all, just looking, you see a whole bunch of different genes and a whole bunch of different epilepsy syndromes. But I'm just going to highlight two examples. I'm highlighting here a chloride channel subunit mutation and a sodium channel subunit mutation, SCN1A, which most of you are probably familiar with. But the reason I'm highlighting these is to show that there is phenotypic variability for these given mutations. So this particular mutation is associated with childhood absence epilepsy, as well as juvenile absence epilepsy and juvenile myoclonic epilepsy. And the SCN1A mutation is associated with genetic epilepsy with febrile seizures plus, severe myoclonic epilepsy, and migrating partial epilepsy of infancy. So we still require much more explanation to be able to say a given mutation causes a given syndrome. One example in which this has clearly been advanced is being able to distinguish mutations in the sodium channel subunit associated with a relatively milder form of epilepsy, which is genetic epilepsy with febrile seizures plus, and a much more severe form, a severe myoclonic epilepsy of infancy, or Dravet. And in that case, the details here are unimportant, but knowing, for example, that the mutations exist in this intercellular region of the subunit is highly predictive of the more severe form of the epilepsy. And in fact, essentially all of these are truncating mutations, which you would predict. But that's the way the analysis has advanced. Here's the list again. And in this case, I've highlighted syndromes that are similar. So in yellow are all juvenile myoclonic epilepsy cases. And in the purple is early infantile epileptic encephalopathy. And in this case, I'm just trying to highlight that different genes can cause a similar syndrome or similar phenotype. And this refers to the concept of locus heterogeneity. So again, there's more that we need to understand to figure out how these mutations in completely different genes can lead to a seemingly similar syndrome. Okay, a couple other comments about the discoveries that have been made to date. A whole bunch of papers on copy number variants. We know that they play a role. An important study by Heather Mefford here in this part of the slide shows that copy number variants, when you look at a fairly large cohort, may be explanatory in 5 to 7% the patients with certain epilepsy syndromes. So that's been an important contribution. Another advance, although it's not too significant, I think most of us would agree, is trying to identify common genetic variants. And so this was a paper that was published just last year. And this is actually the first success using GWAS in identifying potential loci that are associated with the more common forms of genetic generalized epilepsies. And you can see the loci that are highlighted there. This is a neat figure that Ingo Helbig actually came up with. We wrote a paper together, but this was truly all Ingo's work. It's sort of his view of where things have been going with epilepsy genetics. And as you can see, it all started actually way back here in 1989, but then the actual identification of epilepsy genes, beginning with that acetylcholine receptor subunit. And things were getting so exciting over the matter of 3 or 4 years that we were really hopeful we were going to go like this in terms of discovery. And then Ingo calls this region of GWAS the Dark Ages, when nothing much happened. And it wasn't really until we approached the next generation sequencing bubble, as he called it, that things have really picked up a lot. And I hope I'll convince you over the next number of minutes that, in fact, we are really headed, at least for the near term, on a vector that's going pretty quickly upward. So in terms of the way we can think about the causes of epilepsy in 2014, this is again from Ingo. Obviously, they're like in most diseases, like virtually in all diseases. There's this spectrum between genetic and structural metabolic. Virtually all of the main discoveries up until very recently have been purely genetic syndromes that are relatively rare families, Mendelian inheritance, and these would be single gene epilepsies. And then there are the structural metabolic, the otherwise perfectly normal individual who unfortunately has a terrible traumatic brain injury. There may be a slight genetic predisposition, but it's probably very, very minor. And that individual goes on to develop full-blown epilepsy, possibly medically refractory. And then there's everything in between. We believe, based on the data that we've looked at, that there will be a significant number of patients who have a more complex form of epilepsy with a genetic basis. And this is admittedly a skewed view of this pie, but we think that unknown idiopathic is probably genetic and epigenetic. So now for the main part of what I'd like to cover, and that is the next wave. And for this, I want to talk about EPGEP, the Epilepsy Phenome Genome Project, and Epi4K. So a little bit, here's a bit of a story about the project that I hope some of you might find interesting. I want to tell you about how we came up with the idea, because it started actually now 12 years ago in 2002. And for those of, especially for the young people in the room, this is definitely the way some aspects of science works. So a really good friend of mine, Ruben Kuznecki, who at the time was at the University of Alabama, he's a well-known epileptologist whose area of interest is in particularly neuroradiology and imaging as it relates to epilepsy. So Ruben and I are really good friends. We share friendships similar to the friendship I have with Karen and Steve and others. We're a pretty close-knit community around the country and the world. And Ruben and I were talking on the phone way back then, and we were just thinking, what could we work on? What could we tackle that would be of potential interest in terms of advancing our understanding of aspects of epilepsy? And our discussion, it was just a casual discussion on the phone one day, and our discussion was driven in part by the fact that the genome had just been published, the 2 versions in Nature and in Science. And of course, we were aware back then, this is such a old hat story now. I mean, this is like nothing for 2014. But we were aware of what people were predicting with regard to the pace of discovery in genetics. And so all of you have probably seen a figure that looks something like this, which is a prediction of the number of genomes sequenced over time. Not prediction, actually, a tabulation of the number of genomes sequenced starting back in 2000 to 2010, the cost of sequencing, and also superimposes Moore's law, the cost of computing, which has been incredibly predictive with regard to the computational power of an index desktop machine costing $1,000, right? Gordon Moore predicted this back in the late 1950s, early 60s, and it's been essentially correct. Well, what Ruben and I were thinking about back here, knowing that this is what people were talking about, is that the day was going to come when the sequencing dudes were going to come to the epilepsy world and say, you know, hey, we can do a sequence, $1,000, turnaround in a week. Give us your samples. And those of us in the epilepsy world would be standing there and look at one another and say, be right back. You know, we'd be running because we didn't have it. We didn't have these samples collected. At least we didn't know of it in the United States. So Ruben and I started talking about this, and we thought, you know, let's get together a group of people and think about how we could prepare ourselves for that inevitable day. And you know, I remember saying, Ruben, I don't know whether that's going to happen in 20 years or 15 years or 7 years. I have no idea. But the day is going to come when it's going to be absolutely available to everyone to be able to do a whole genome, a whole exome, or whatever. So we had a meeting. We got together a group of people, again, a whole group of friends, at the American Epilepsy Society meeting in 2002. And we presented this idea. These are all folks who we knew would potentially be interested. We knew them personally. Again, they were friends. They were experts in the field. And we proposed this general idea, and everyone came on board and said, let's figure something out. It took, 2002. It took 4 years to actually get the grant, 2 years of planning, a year of solace as we got denied, and then eventually the grant came through. And this led to the Epilepsy Phenome Genome Project. So the goals of the project have been to create a database of very, very well phenotyped patients with epilepsy, specific forms that I'm going to tell you about in a moment, to create a repository of DNA and cell lines for all the participants, and actually have them available to investigators anywhere in the world who might be able to benefit from analyzing that material, to look really carefully at the phenotypic characteristics of this cohort, to look at potential phenotype-genotype relationships, and then eventually to apply the state-of-the-art genetic techniques to analyzing the data set. NINDS, when we first went in, we actually asked for the whole thing. We asked for everything for collecting the phenotype data, collecting the patients, banking the DNA, as well as the genetic analysis. At the time, it was GWAS. I think the original budget was $28 million plus indirect. NINDS, or the review committee, very wisely said, you know, if we're going to fund you, we'll fund you for collecting the material. We'll hold off with regard to the genetic analysis. Really, really smart. Of course, we were unhappy for a couple of months, and then we put in a revised proposal and got the funding. I think the original grant was about $15 million plus indirect costs. So here's what we've been working at since 2006. Our goal has been to collect two main groups of patients. The first group are individuals who either have idiopathic generalized epilepsy, so unexplained epilepsy without any obvious acquired abnormality. Today we would call this genetic generalized epilepsy with the new International League Against Epilepsy nomenclature. Patients with IGE who had a first-degree relative who also had epilepsy. It didn't have to be IGE, though. It could also be localization-related epilepsy. So patients with focal seizure disorders, again, without any obvious cause. So an MRI scan had to be obtained to rule out any developmental abnormality, old traumatic brain injury, old stroke, evidence of infection. It would be a cryptogenic form of focal epilepsy. So in this, for this group, we wanted either IGE or LRE probands who had a first-degree relative. So that could be a parent, a sibling, or a child. And we were seeking 750 of these pairs. The second group had quite a different form of a set of epilepsies. These were individuals who had either infantile spasms, Lennox-Gastaut syndrome, or malformations of cortical development. They were the probands, and in a few minutes I'll explain the rationale for why we collected these patients this way. But in this case, we wanted to make sure that we could get the DNA on both of their parents who were normal. So these were trios or triads. And as you can see, we sought to collect 250 of each of these. These two, infantile spasms and Lennox-Gastaut, I'll refer to in a few minutes as epileptic encephalopathies, because these are more severe forms of epilepsy that have with them significant cognitive delay or decline. So the total was 5,250 individuals, of whom 3,750 had these various forms of epilepsy and 1,500 parental controls. Here's how we went about doing this. I won't bore you with all the details. I just wanted to mostly make the point that it was a lot of work. It's been a lot of work. We decided at the beginning that we wanted to be absolutely sure that we had the diagnosis correct, because having any false positives in this database, that is, having a DNA sample that was labeled as someone with epilepsy who actually turned out to have another disease, will really mess up the analytics. So we put an enormous amount of work into figuring out the protocols for doing epilepsy phenotyping. And for any of you who have done this before, you know what I mean when I say it is not easy to do phenotyping of patients with epilepsy. But we had a whole a set of processes. I'll just emphasize this one part. To phenotype the individuals, there was a structured diagnostic interview that was done person to person or over the phone. We abstracted information from the medical records. There were a whole slew of supplemental forms. We attempted to collect information on anti-epileptic drug use, but actually we had to pitch that after about a year and a half because it was just too difficult to do it retrospectively, to our great disappointment, but it was a sort of a realistic choice. And then we had a series of cores. The EEG core reviewed every EEG that was submitted. An MRI core led by Ruben looked at every MRI scan to make sure that the local neuroradiologist had not missed some important lesion and so forth. And then we actually had a review to make sure that there was consensus around the final diagnosis. Here, this is what our informatics infrastructure looked like. Again, the details don't matter. It just was a big investment of resources and time to be able to have a database that actually today I can go on our database and look up an EPGEP patient using a Google-like search function and review the details as they relate to a gene that we've recently been sequencing. Again, just to emphasize the approaches that we've taken, and I'm more than happy to share any and all of this, these approaches and the experiences that we've accumulated with anyone who's interested. But in order to finalize the final diagnosis, we had a review core, a data review core, in which a case would be randomly assigned to two reviewers who would independently review it. If there was no concern found about this review, it was considered complete. If there was, there'd be a primary reviewer. The concerns would be sent back to the site. They would have to respond to it. We'd have to achieve consensus, and it would finally be complete. EPGEP relied on 26 centers around the world, most of them in the United States, distributed as you can see here. But because in the first year or so, year and a half of recruitment, we weren't meeting our recruitment targets, we scrambled to figure out how to deal with this. It's a common problem in any kind of clinical study. And we actually created a community physician referral network distributed all around the country, which really helped a lot in terms of getting closer to our targets. And anyone who was at the American Epilepsy Society meetings in 2007 and 2008 were probably accosted by some of the EPGEP investigators, giving you little pins and coffee mugs and stickers and things to remind you about the study. So here's what we've accomplished. This is as of October. Actually, the study enrollment ended then. And as you can see, the target that I mentioned before was 5,250 individuals. We actually consented over 5,500 people. There was dropout for various reasons. Some were ineligible. For some, we couldn't get DNA samples. And our final target we achieved was 4,185. We fell short primarily on the localization-related epilepsy patients. They were just harder to find in terms of having a first-degree relative. This represented almost 34,000 activities, and we have about 7 million data points in the data warehouse. A number of, as I said, one of our goals was to look at just the phenotypic characteristic of the patient populations, and there have been a whole number of papers that have come out related to that, including observations with the malformations of cortical development, some new observations about Lennox-Gastaut syndrome, an interesting study looking at racial and ethnic differences, and a paper that I'm just going to highlight for a moment. There we go. The paper by Melody Winnower and others looking at shared genetic susceptibility to migraine. Just one example of some of the things that can be teased out when you have a large, carefully phenotyped population. So the group, the patients actually all went through an interview that asked about a headache history because we were interested from the beginning as to whether or not the association that's been described between migraine and epilepsy might exist in our cohort. So they all did this interview, and then they could be split into patients who had migraine with or without aura. And these are the main findings of the study. And that is of a group of 730 individuals with either idiopathic generalized epilepsy or localization-related epilepsy, that in fact there was an increased likelihood of the members in the cohort of having migraine with aura that correlated with the larger number of family members with epilepsy. And that's shown here in terms of the odds ratio, and that is that migraine with aura had an odds ratio of 2.5-fold increased likelihood of finding in the family. It was not seen in regular migraine. That's kind of interesting because migraine with aura is associated with cortical spreading depression, and one might think that the types of abnormalities of network excitability that's associated with epilepsy would also be seen in migraine with aura, and again, from a genetic basis. Okay, so now let me turn. That's a bit about EPGEP, and now let me tell you a little bit about Epi4K. So this was, the idea of Epi4K came in large part, as a response to a request for applications from NINDS, looking for funding or offering funding to a center without walls that would focus in on the genetics of human epilepsy. And it was this group of people that got together, again, group of people who knew each other, different fields of expertise, liked working with one another. And we put in a proposal, and we were very, very fortunate now 2.5 years ago to get funded. And this is the way Epi4K looks. Epi4K stands for epilepsy sequencing of 4,000 genomes. And this is the structure. We have an administrative core. We have a core that focuses in on the phenotyping and the clinical informatics related to the cohorts that are studied. And then we have a sequencing core that handles the actual DNA sequencing and the analytics. This group is led out of UCSF, but has a number of different centers around the world. And the sequencing core is really David Goldstein and his group at Duke. And then we went in suggesting 4 projects. Project 1 is entitled Epileptic Encephalopathies. I'm going to come back to that in about 4 or 5 minutes because that's the main results that have come so far. We have a second project called Multiplex Families and Pairs, in which we're looking at cohorts similar to the IgE-LRE cohort that I described for EPGEP, the idiopathic localization relateds. There's a prognosis core that wasn't funded that we're going back in on, and then a 4th project on copy number variant detection that's being led by Heather Mefford and Evan Eichler at the University of Washington. So before I tell you about Project 1, a little bit more about the way we organized ourselves, because I think it might be instructive. When you bring together large groups of people like this to work, we found through our experience with EPGEP that it was enormously important to think about the ground rules, about the nature of the relationships between members of the team. How exactly were we going to make sure that we were all on board working collaboratively? I mean, EPGEP had over 100 investigators, plus study enrollment personnel. So we had a huge number of people working collaboratively. We needed to figure out the rules. So we needed to come up with sort of a contract or a memorandum of understanding. And let me just show you a couple of those details because I think the message will be pretty clear. First of all, how do you make sure that people are, in fact, working collaboratively? So we actually have a charter. This was based on the EPGEP charter, but I'm showing you the one for Epi4K. And let me just actually let you read it, just to give you a sense of the kind of language that people use. Okay, so that gives you a bit of a taste. I just wanted to emphasize, this is all about how so much more can be accomplished when you work together rather than work competitively. And also, this one. This has really been important. So when you get a large group of people, which include very talented investigator groups that are doing their own work in the genetics of epilepsy, you can't tolerate the possibility that on a steering committee phone call, someone says, hey, by the way, Dan, I heard that your group was working on, did I get this right? Doesn't that sound like it's like in direct competition with what we're doing in Epi4K? No, that's a surprise, right? So this principle of no surprises has come up many times. The other thing I wanted to emphasize is you can imagine that maybe authorship has a bit of a complexity in this type of work. Right. So here's the way Ruben and I actually originally thought about this with that fateful meeting in 2002 at the American Epilepsy Society. We conjured up an approach and presented it to the group. And the deal basically was this. For any principal paper, any primary paper that comes out of this collaborative work that we do together as a group of however many investigators, the deal is if you're a full professor or the equivalent, for any primary paper, you will not be a named author on the masthead of a paper. And that's the deal. It was interesting. Only one person left the room. And we probably didn't really need him, right? So this is what our publication policy looks like for Epi4K. And so, again, there's a sort of intro that says there's an agreement that we all embrace the concept of collaboration when we think about authorship. You have to contribute directly to this work, and we'll share in the credit. And in fact, investigator status in Epi4K signifies a clear acceptance of the policy that all main scientific results, which are the primary papers, which are the things that we predict we're going to be able to write on, will be attributed to the sole author of the Epi4K consortium. And everyone, you have to sign on. So we did that. But in addition, we recognize that there are hopefully going to be plenty of other papers, and we want to make sure that the junior investigators that get the opportunity to take advantage of that. So in fact, we also say that priority is given to junior investigators for named authorship. So you probably saw from the EPGEP papers that there were authors listed on the masthead, but every one of those named authors were assistant or associate professors or other members of the lab. And the rest of the senior investigators were listed as the EPGEP investigators. This is still authorship. You still can find your name in PubMed, but it's the way that we've dealt with who's going to be first, who's going to be last, who's going to be corresponding. It's been incredibly effective in maintaining the cohesiveness of the collaboration. Okay, so I'm going to finish. It'll probably be another 5 minutes or so, Karen. Is that 5 to 8 minutes? Okay, we're good for that? Okay. So what I want to finish and tell you about the first main success of EPGEP and Epi4K. And I'm going to add in just a couple of things that have happened since then, which I find very intriguing. I hope you will as well. So I mentioned that Project 1 is called Epileptic Encephalopathies. Now, the epileptic encephalopathies, I've already mentioned these two categories. They include infantile spasms. These are individuals, people born early in life who have the onset of their epilepsy, a particular type of epilepsy or type of seizure called infantile spasms, between 4 and 12 months of life. The EEG has a very characteristic finding called hypsarrhythmia. More than half of the infantile spasms cases have something identifiable from a structural point of view, or a clear acquired lesion, developmental brain malformations, or clear chromosomal abnormalities, which have been known for some time. And they may evolve into this other syndrome called Lennox-Gastaut syndrome. These are patients who typically develop refractory epilepsy and have cognitive decline, very, very severe forms of epilepsy, many different seizure types, significant intellectual disability appearing during childhood, and then they develop into adults with no improvement. And again, in this case, about a third of Lennox-Gastaut cases have no obvious cause. So, in designing the approach, the original approach for in EPGEP, where I highlighted that we were looking for probands in both normal parents, the hypothesis was that these individuals could well have some kind of newly acquired genetic abnormality or de novo mutation. And the reason behind that is the following. Most epileptic encephalopathy patients don't show transgenerational inheritance. Most families don't have sibling recurrence, and so that's our hypothesis, that infantile spasms and Lennox-Gastaut patients have de novo causal mutations. And in order to get a high yield of these types of individuals, we targeted families in which both parents were available, no epilepsy in the parents, and no familial recurrence. And we excluded patients with known causes, hence the requirement for imaging to rule out, say, an undiagnosed case of tuberous sclerosis or any other kind of acquired lesion. And this is the result of the work. It's a paper that came out in Nature back in September, and I'll tell you just a little bit about the results. We had 264 probands that underwent whole exome sequencing by the Duke lab, and both of their parents, so something on the order of 770 or so exomes total. And I won't go into the details of the analysis that the Duke group used, but what they did all this. Most of us didn't have anything to do with the exome analysis and the statistical approaches that were taken. But what was discovered is that there was an excess number of de novo mutations in the epileptic encephalopathy probands. The average number of de novo mutations that any of us walk around with is about 1.1. And so you can see that there were a number of individuals, a significant number of individuals who had 2, 3, 4, or more de novo mutations in their exome. Now, a quick side story. So this is a picture of Slave Petrovsky. He works with Andrew Allen at Duke. And Slave is a young statistical geneticist who became involved in Epi4K because David and his group was part of the sequencing core. And Slave, as part of that group, became interested in what we were doing. Independently, he had been thinking about new approaches of analyzing the occurrence of de novo mutations in the genome. And he and Andrew posed a really interesting question, and that is, given that there are a certain number of de novo mutations that occur just randomly, by chance, during oogenesis or spermatogenesis, which we then acquire at a rate of about 1.1, if you think about where the mutations are distributed across the entire genome, or across the exome, where the genes are they randomly distributed, or do they tend to fall into pre-selected or somewhat selected groupings of genes? A really interesting question. And I'm not a geneticist, so I don't know the history of how that question has been asked. But apparently, it has not been asked in a very, very quantitative way. So Slave, actually using existing databases of the exomes that have been reported, the known de novo mutations that have been associated with diseases of all types, and looking at their distribution across the known human exome, discovered that, in fact, it is not distributed by random. And, in fact, there are genes that you could call relatively tolerant to de novo mutations and genes that are relatively intolerant. And they go across a spectrum. And this is a graph that shows the distribution in terms of the sum of all the variant sites in a gene and all the common functional variants. And the highlighted in blue are the extreme 2% of the genome that are relatively tolerant to de novo mutations, meaning that they don't tend to cause disease, as opposed to those that are very intolerant. And I'll just highlight a couple to make this point. Here's the HLA antigen group, right? Well, nature designed those genes to be quite tolerant to mutations because that's part of the way the immune system works. Similarly, olfactory receptors, they're designed also in order to have the spectrum of chemosensitivity by having these random mutation events. Compare that to calcium channels, right? Not very tolerant at all, given how much they control excitability of, say, the heart and the brain. So with this type of approach of analyzing the genetic landscape of de novo mutations, the group was able to apply this to the de novo mutation gene set that was discovered in the 264 probands. And what we found was that, in fact, the epileptic encephalopathy de novo mutations in our cohort, as well as what was published in the cohorts with intellectual disability, actually have an overrepresentation of de novo mutations in the more intolerant genes, adding to the likelihood that they are causal with regard to disease. Does that make sense? Okay, good. And this was significant. Interestingly, what had been published on autism spectrum disorder, including those 4 papers in Nature more than a year ago, did not meet that standard. Here are the main discoveries. These are individuals who we discovered de novo mutations in identifiable genes that all except 1 or 2 can be said to be causal. The ones in red, actually, there had been individual or small series reported previously. So, for example, no surprise, significant number of patients with SCN1A, even though we tried to rule out patients with a phenotype that was obvious for SCN1A. But these included this list, as well as 2 new genes, the GABA3 receptor and ALG13. Saw 2 patients in one case and 4 in another, again, achieving high significance. So these are genes in which we are quite sure are causal of disease. There were a whole number of other de novo mutations in which there was only 1 individual identified, and so we can't yet say for sure that they're causal in the EPGEP cohort, but there are certainly reasons to believe that they would be because they've been identified with a whole variety of neurological syndromes previously. And it was interesting that if you look at the de novo mutations overall, that they fall into particular gene sets. So a significant overrepresentation of genes that are among the ion channels, genes that have been already associated with monogenic disorders, so those rare families, that long list that I showed you before, autism spectrum disorder, intellectual disability, and Fragile X. And here's a pathway analysis. I won't go into the details here. This is a very coarse method of looking at protein-protein interactions, and the findings here really were, again, an overrepresentation of the genes in this network, and they do, again, fall into this group of both this particular network has an overrepresentation of genes that have been identified in epileptic encephalopathies, autism spectrum disorder, and infantile intellectual disability, and Fragile X. So I'll bring it to a close. I'm just going to tell you just a couple more things. So here's what we're planning to do with Epi4K and EPGEP. More analysis of the phenotype data set. Whole exome sequencing has been done on an additional almost 100 epileptic encephalopathy trios in collaboration with European colleagues. And I can't tell you the results yet, but we've discovered some additional causative variants. We've done whole exome sequencing on the malformation patients and have some interesting results. We've actually done whole exome sequencing on the first 900 EPGEP patients, that first group that I showed you, that we're collecting with IgE and LRE, and again, interesting results that we're just looking at over the last couple of weeks. And then we're beginning to do functional analysis of some of these variants. 2 really quick things. And that is, I'm sure most people are aware of Scott Baraband's work screening zebrafish with that have the SCN1A mutation genetically introduced into the zebrafish line. And again, I'm showing this mostly because it's an example of how the identification of a causal variant can lead to actual therapies. So what Scott has done is he's created a really, really neat monitoring system in which he can take these variants that have been introduced in the zebrafish and can place them into 96 well plates, and using a video system can identify the seizure type phenotype. So here, this is the zebrafish that has the SCN1A mutant, and it's basically swimming all over the place because it's having seizures. This is what a control is like. And he can do forebrain electrophysiology and directly measure the activation. I should say this work was fundamentally funded by Cure. It probably wouldn't have gotten off the ground had it not been funded by Cure initially, because NDS apparently was not interested early on. Anyway, Scott was able to actually take a drug collection that had something like 325 different compounds and very rapidly screen them, and was able to actually show that the known drugs, these are all various types of anticonvulsant drugs, many of them actually did have a significant effect on the seizure phenotype. And then he did the drug screen, and those shown in red were considered significant in terms of meeting the threshold for reducing the activity. Ones that are right down at -100 were toxic. These are animals that were dying from the medication. But clemizole is this very interesting H1 antagonist that came out from the screen that's already been approved by the FDA back in the 1950s and 60s. So a really cool example of how knowing the target can lead to a potential therapy. And I'll just mention one other. This is a paper that's in press that Carol Milligan, Steve Petro, and colleagues are publishing. And this is taking a look at KCNT1 and discovering a compound that seems to be effective, at least in vitro. So it turns out that the KCN, this potassium channel mutation, is associated with a severe form of autosomal dominant frontal lobe epilepsy, which is one that more typically involves the nicotinic-cholinergic receptor. But this is a more severe form, generally earlier onset, associated with intellectual disability, and as I said, associated with this particular mutation. There's also this thing called epilepsy of infancy with migrating focal seizures, which is a very severe form of epileptic encephalopathy that has some similar characteristics, but overall is more severe than the severe form of autosomal dominant frontal lobe epilepsy. So what Carol did was take these specific mutations in humans and introduce them into oocytes and measure their effect. And what she found was a dose response in terms of the influence of these various mutations on current associated with increased severity. And they turned to quinidine because quinidine is known to reverse the effect or actually change the effect on potassium channel currents, even in the wild type. So she reproduced that effect in the wild type, but then applied quinidine to specific mutants that are associated with these various forms of either the severe form of ADNFLE or the epileptic encephalopathies, and in fact sees a partial rescue effect. Again, a drug that's available, can be used. It's going to go into clinical trials. So I'll now conclude. I hope I've gotten across the point that living with epilepsy is really an incredibly challenging experience for both the person with the epilepsy as well as family members and others associated. And again, I can't do justice to the frustration that I see in the eyes of the patients that we take care of and their family members, and the frustration that we feel as healthcare professionals in attempting to try to bring some answers to the three questions that I mentioned before. I think because of the progress in next-generation sequencing and the way the groups are working together, that we're on the upswing in terms of understanding some of the genetic architecture of the epilepsies. Large-scale collaborative efforts are absolutely essential to this. There is no, in fact, during lunch, I forget who said it, one of the graduate students independently talked about how our future is so much dependent on working together in teams. Someone said, you know what, you can't even go in with an NIH grant without showing that you've got the spectrum of capability, the versatility, the training to be able to take on these large questions. And certainly one aspect of science is going to continue to be driven that way. Just to make this point, here's just a partial photo montage of the people who have been involved in Epi4K and EPGEP. And I sent them a survey now about a year ago because I was just interested in trying to document what the scope of training was among something like, 143 people, right? So I asked them, tell me what kind of postgraduate training you've had. So it doesn't include master's degree. Actually, I did include master's degree, but anything beyond your regular undergraduate training. And as you can see, here's the distribution, a whole bunch of MDs and PhDs, and here's all the different areas that people got degrees in. So that's the expertise, the scope of expertise that's represented when you bring together a group like this. And oh, by the way, this is 991 person years of training. So we almost a millennium. Finally, the results that groups from all around the world are having in the genetics of epilepsy are having a real impact on patient care. For the pediatric epileptologists in the group, you know that when we talk to families now, being able to get some answers with regard to the genetic cause of the disease, just having a potential answer can have a huge impact in terms of thinking about the future of that child, thinking about family planning and so forth. And I think that the latest results that we've gotten in the world of epileptic encephalopathies suggest that we're going to be able to identify the causative variants in these individuals. At the moment, we have explanations for about 12% of the epileptic encephalopathies, the pure epileptic encephalopathies. We're quite sure that that number will be on the order of 25 to 30 to 40% just based on having a large enough cohort. So once we get to 1,000 or 2,000 individuals, we'll start seeing double hits and triple hits in some of those individual variants and be able to do the statistical test to determine whether it's causative. And fortunately, it looks like they may be falling into different groups, which has therapeutic implications. And finally, just highlighting the work that Carol Milligan and Scott Barabin have been doing, these types of functional assays and high-throughput screens are really producing some surprising findings. Whoever would have thought that clemizole was going to surface, or quinidine, but it's probably just the beginning of many discoveries of that kind. We really welcome your participation. EPGEP, we have a database that's becoming publicly available, so if you're interested in looking at it, it's yours to enjoy. All of the DNA that's been collected for EPGEP is deposited at the Coriell Cell Repository. If there's not DNA, there are cell lines, so there will be a source in perpetuity. All the results that come out of EPGEP and Epi4K are placed in dbGaP, so the data from the Nature paper is already in dbGaP, so it's there for you to look at. And if you've got any interest in collaborating, we get actually a fair number of letters of request for collaboration in which laboratories or groups are looking at the opportunity for melding together data sets or looking for whether there's a potential functional variant on a particular molecule that a lab's interested in. So we welcome your involvement. And with that, I'll say thank you.
Thank you very much, Dan. That was outstanding and really informative. And certainly we have some time for questions if people are interested. We run a little bit over, so if people need to go, we understand that. But certainly the floor is open for some questions for Dan. And I might start with one of my own. Dan, so what you've described is a really incredible system to be able to discern the mechanisms underlying neurological disease for epilepsy. And obviously there's a lot of ways you could apply this to schizophrenia or any other neurological or any disease, really. Has this approach been begun to be adopted by other groups?
Yeah. There are lots of groups around the world that are working on you name the disease and people have organized. So the multiple sclerosis, clan has gotten together. I think I don't think there's anything special that we've done other than what I emphasized about the charter and the publication policy. That's been met with a fair degree of discomfort. And so I think that the groups that are say that it's hard to do this are the ones that haven't made the a little bit difficult but important upfront decision about going this way. Now, related to this is if the University of Utah is similar to UCSF, you probably are not as advanced as we all should be with regard to recognizing the contributions of individuals in large scientific enterprises. So, for example, the wording of your promotions criteria probably don't sufficiently pay attention to what you need to demonstrate in order to become an associate or a full professor. But that's just simply got to change, because I can tell you from firsthand experience, and any more senior faculty member here, I know you'll say the same thing, is when we look at the promotions packet for an individual, we're just it's almost a habit to look at the list of publications, sort of count them up, but in particular pay attention to where you're first or last author. And if you're in the middle, it just doesn't count as a sort of primary paper. So this is a big problem, because I just showed you an approach that we're taking in which there aren't first or last authors, at least for these larger, higher impact publications. The solution is to change the way we provide the details on what exactly the contribution was of an individual. And you've already seen this in some of the journals, where there'll be a list with each author describing what it is that the individual did. So what we would like to do is have the journals adopt a framework in which there's basically a table. And the table has, in the first column, the list of all the authors, alphabetically or reverse or randomly, whatever. And then the columns are the different activities that the individual is involved in. So, for example, in the Nature paper, we actually put in a table, but Nature forced us to use little superscript numbers. It's impossible. But the table was much simpler. And the first row has enrollment of patients, phenotyping, exome analysis, statistical genetics, writing of manuscript, principal investigator. And then there's just a check mark for each activity that you're involved in. And so as a member of a promotions committee, I think we should be looking at that level of information and say, oh, this person participated in a high-profile paper, but her involvement was enrolling patients in phenotyping. This individual wrote the that's the way I think that these things should be ascribed. Yep. Any other questions? You've got to have something. Yeah.
You presented data for both the autism spectrum disorders and for migraine spectrum disorders. Did your database collect information on any of the other neuropsychiatric comorbidities?
Yeah, so we don't have the autism data. I referred to some prior papers, but we do not. Actually, we anyone with a more severe form of autism or any kind of pre-existing developmental delay they were not included in our cohort. Yep.
Okay.
Okay.
Thanks
Good.
so much, Tim.
Thank you.